And good evening, ladies and gentlemen. Welcome to D.G. Wills Books in La Jolla, California. This evening, it is our great honor to host Dr. V.S. Ramachandran, who is here this evening to discuss his new book, The Telltale Brain, A Neuroscientist's Quest for What Makes Us Human. At this point, I'm also honored to turn the proceedings over to Roger Bingham of the Science Network.
So thank you, Dennis, and good evening to everybody. One thing, we are filming this, as may be obvious from the cameras, so if anybody's here is on the lam or is here with somebody they shouldn't be with and don't want to be seen with them, good moment to leave at this point. The Science Network, by the way, if you go and look at www.thesciencenetwork.org, you will see that we have a huge collection of interviews and conversations on there, plus we do meetings like the Beyond Belief meetings that were done at the Salk Institute. Does anybody know what's special about today? Do you know what's special about this particular day?
It's her birthday.
Well, you share a birthday with Francis Bacon, who in 1561 was born this day, 22nd of January. Francis Bacon obviously became Lord Chancellor of England, wrote the Advancement of Learning, Novum Organum, and the wonderful novel, The New Atlantis. And his work essentially became the model for the setting up of a community of scholars, which became the Royal Society later in 1660, which was their 350th anniversary last year. So his 450th birthday is today, and I would like you to say, happy birthday, Francis Bacon. Thank you. That's really kind. What was great about Bacon, some of the things that Bacon wrote, is that he talked about an island of Ben Salem and a place called Salomon's House. And he had a community of scholars there. And what they dealt with was one commodity only, which was light, the light of understanding. And those people he called merchants of light. And I'm going to suggest to you this evening that Ramachandran is in fact a merchant of light. And I'd like to say a little bit more about Rama. He's obviously, he's here at UCSD, director of the Center for Brain and Cognition. He's a distinguished professor with the Psychology Department, Neuroscientists program. He's extraordinarily well known for doing these immensely inventive investigations about people. And I think I want to read just a tiny bit from the preface here, because it gives some sense of what Rama does. My preference for low-tech methods has both strengths and drawbacks. Of course, I enjoy it, partly because I'm lazy, but it isn't everyone's cup of tea. Most individual researchers need to specialize, but the scientific enterprise as a whole is made more robust when scientists march to different drumbeats. Homogeneity breeds weakness, theoretical blind spots, stale paradigms, and echo chamber mentality, and cults of personality. A diverse dramatis personae is a powerful tonic against these ailments. Science benefits from its inclusion of the abstraction-addled, absent-minded professors, the control-freak obsessives, the cantankerous bean-counting statistics junkies, the congenitally contrarian devil's advocates, the hard-nosed, data-oriented literalists, and the starry-eyed romantics who embark on high-risk, high-payoff ventures, stumbling frequently along the way. If every scientist were like me, there would be no one to clear the brush or demand periodic reality checks. But if every scientist were a brush clearing, never stray beyond established fact type, science would advance at a snail's pace and would have a hard time unpainting itself out of corners. Getting trapped in narrow cul-de-sac specializations and clubs whose membership is open only to those who congratulate and fund each other is an occupational hazard in modern science. Rama is not one of these people, as you probably know. He's an extraordinary, inventive investigator. I want to emphasize as well that although you'll find in this book many of his signature investigations into extraordinary conditions, like the phantom limbs, like Capgras, like the Cotard syndrome, like apotemnophilia, get him to ask you what that is, and his discussion of mirror neurons, which he's been championing for a while now. I actually don't want to go into that because he's going to do that, but I do want to make another point, that throughout the book you'll also see that he has a number of wonderful sort of two cultures references. It's a very literary book, and I wanted to sort of mention to you he has a quote in there from, we both do an awful lot of Shakespeare references in what we're doing. And I wanted to mention something here which will give you some clue of the kind of territory that Rama goes into. This is Act 5, Scene 1 from the A Midsummer Night's Dream, which I happened to pick up from Dennis's bookshelf when I arrived here. More strange than true, says Theseus, I never may believe these antique fables nor these fairy toys. And here's where it gets interesting. Lovers and madmen have such seething brains, such shaping fantasies that apprehend more than cool reason ever comprehends. The lunatic, the lover, and the poet are of imagination all compact. One sees more devils than vast hell can hold. That is the madman. The lover, all as frantic, sees Helen's beauty in a brow of Egypt. The poet's eye in a fine frenzy rolling doth glance from heaven to earth and earth to heaven. And as imagination bodies forth the forms of things unknown, the poet's pen turns them to shapes and gives to airy nothing a local habitation and a name. There is, in what Rama does, a giving of things unknown, these diseases I've been talking about, these conditions. He turns them to shapes and gives to airy nothing a local habitation and a name. And I think you'll find this amazing to hear what he has to talk about. Finally, if you look on the book, you'll see that Rama has been compared to many people. Richard Dawkins called him a latter-day Marco Polo, journeying the Silk Road of science to strange and exotic cafes of the mind. Eric Kandel said that he's in many ways the modern Paul Broca. Alan Snyder said that he's the Galileo of neurocognition. Norm Deutsch talks about him in extraordinary terms. And all of these folks, although I agree with them, I beg to differ in one sense. I would not want to say that Rama is like the something of something. He's sui generis. He's one-off. Rama is Rama. And here he is.
Thank you very much for coming. And let me begin by, first of all, of course, thanking Dennis for inviting me to give this presentation. I always tell Dennis that this is a tremendous place, this bookstore. It's a little oasis of culture and learning amidst this whole cultural desolation that we call San Diego and La Jolla. And I'd like to thank Roger for this wonderful introduction he gave. But I can continue like that, but I'm going to dive in straight because of the time limitations, talk about the book. And the book, as you know, is about the human brain and studying its functions and structure, which is what I do for a living at the UCSD. And just by way of introduction, as you all know, you may have heard that the brain is an organ of staggering complexity. It's made up of 100 billion neurons, which are the structural and functional units of the nervous system. And each neuron or nerve cell can be either act on or off, and it makes synapses with or points of contact with 1,000 to 10,000 other neurons. And each point of contact or synapse can be either on or off, can be inhibitory, can be excitatory, and somebody has figured out from this that the number of possible brain states, permutations and combinations of brain activity, exceeds the number of elementary particles in the known universe. So given this complexity, how do you even begin to study the functions of the brain? Well, there's several approaches, but before I go there, let me just begin with a statement, my opening paragraph in the book, just to give ourselves a sense of perspective. Here is a lump of jelly, a 3-pound lump of jelly you can hold in the palm of your hand. And this lump of jelly can contemplate the vastness of infinity, vastness of interstellar space, meaning of infinity, meaning of zero, the meaning of God, and even contemplate itself contemplating, and what we call self-awareness. So how does all this come about from this lump of jelly? And this truly is the greatest of miracles. So there are many approaches to this problem of understanding the brain. One is to do brain imaging, ask the subject to do something, perform some task, and do brain imaging, find out what part of the brain lights up when you perform the task. So when I wiggle my finger, what part of the brain lights up when I wiggle my finger? And that is all over the newspapers, and you know, you've all heard of brain imaging. Second approach is to put electrodes in the brain and record the activity of single neurons and nerve cells. And you can eavesdrop on the activity of nerve cells and ask yourself, what is the function of the nerve cells? For example, if you put an electrode in the back of the brain, in the visual areas. If you make loud clapping sounds or wave your hand up and down, the cell won't do anything. But if you present a bar of light, the cell will fire. And this gives you some confidence in saying that cell is involved in processing visual information primarily, but not sound. And you can do this all over the brain. The approach we are involved in at UCSD is called behavioral neurology or cognitive neuroscience, which is you look at patients, neurological patients who have sustained an injury, usually a small stroke or a tumor or a head injury affecting a very tiny part of the brain. And very often what happens is you get loss of one, highly selective loss of one specific function, with other functions being preserved intact. This gives you some confidence in asserting that this region of the brain is specialized for that function. In other words, when there's damage to a small region of the brain, what you get is not an across-the-board blunting of all your mental capacities, but you get a highly selective loss of one specific function. For example, you may get a patient with bilateral damage to an area called MT. This patient can read a book, can name colors, can walk, can talk, can think, can converse fluently. Everything is normal, but he can no longer see moving objects. He sees the object changing position. He says, it's here, but I don't see it moving. He's terrified of crossing the street because he says he can't see how fast the object is approaching. So this gave rise to the idea that this area MT is specialized just for seeing movement, not just for vision, but even within vision, specialized for just extracting motion signals. Similarly, there's an area called V4 for color. So this is the so-called localizationist approach to brain function, and my book is full of examples of this kind. Overall, what we've done is there are many neurological syndromes of this kind which are bizarre or spooky at first, seem bizarre, and have been ignored by the mainstream neurology establishment because you couldn't make any sense of them. They were regarded as anomalies, what Thomas Kuhn used to call anomalies, something that didn't fit the big picture of science, a big picture of neurology. What we've done is to rescue this phenomena, these anomalies, from oblivion by bringing the patient from the clinic to the laboratory, doing intensive experiments on these patients, which allow you to draw conclusions about not only about his maladies, about his symptoms, but also how the normal brain works. So that's the main goal. So let me go through all of these examples are in my book, but just to give you an idea, I'm going to give you a few examples. One is what's called the Capgras syndrome. It's there in my previous book. So about 2/3 of these syndrome I'm going to talk about are new to this new book, but some of them are described in my previous book, Phantoms in the Brain. Capgras syndrome is one such example, but I'm going to go over it briefly anyway. Initially I was supposed to show this using slides, but somehow we didn't bring a slide projector, so that's not going to happen. So on the sides of the brain you got the temporal lobes, and tucked away in the inner surface of the temporal lobe is a structure called the fusiform gyrus, about the size of my pinky, my little finger, which seems to be concerned primarily with processing faces. It's got other functions too, but one of its main functions is processing and recognizing faces. And when this is damaged, you get a patient who has face blindness. He can no longer recognize people by looking at their faces. Now, he's not literally blind because he can see objects, he can name objects, he can read a newspaper, he can name the color of a car, but he can no longer recognize faces by looking at the features. Now, you can still see a face because you can show him a face and give him a multiple choice of faces, and he'll pick the correct partner for that face. So you can see all the features and everything. What eludes him is the identity of the person he's looking at. Is this Joe? Is this Harry? Is it Susan? Is this face familiar? Where have I seen this person before? Is it somebody recognizable? Is it somebody I should watch out for? All of those things are missing. So identity is missing. Now that's a well-known disorder, has been studied many times by cognitive psychologists. But another disorder which we have studied, which is much more rare, is called the Capgras syndrome or Capgras delusion. This refers to patients who have had a head injury, sometimes, or a stroke. The patient, like a patient I saw not too long ago, was in a car accident, was in a coma for a month. He was actually a student of mine. Came out of the coma, seemed quite normal, was verbally fluent, articulate, intelligent, emotionally not disturbed in any way. I'm going to switch this off. Emotionally intact, but he had one profound delusion. He'd look at his mother and say, "Doctor, this woman looks just like my mother, but in fact she's some other strange woman pretending to be my mother. She's an imposter." This is the so-called imposter delusion or Capgras syndrome. There are many case studies in the literature. But in fact it's quite rare and very few neurologists have heard of it. So here's a person who can see that it's his mother. He says, it looks like my mother, but this delusion kicks in and he says, but she's not my mother, she's an imposter. Sometimes it affects a husband or a spouse or a child, but very often it affects a mother. Now, why would this happen? Now, the standard explanation for this is Freudian, believe it or not. And the idea is this chap, when he was a little baby, and the same argument applies to women, but I'll talk about the guys. When he was a little baby, a little infant, he had a strong sexual attraction to his mother. I'm not saying I believe this, but this is a standard Freudian view. It's called the Oedipus complex. Okay? Then as the brain of the child develops, the cortex develops, and then it inhibits the latent sexual urges. Thank God, otherwise we'd all be sexually aroused by seeing your mother. And then what happens is you have an accident, there's a blow to the skull, an injury to the cortex, and there's inhibitory signals from the cortex to the limbic system, the emotional centers, is removed. The inhibition is removed, so all these latent sexual urges, Oedipal urges, come flaming to the surface, and suddenly and inexplicably you find yourself being sexually turned on by your mother. And the only way you can make any sense of this is by saying, doctor, this is not my mother, this is some other woman, she's an imposter. It seems very bizarre to you and me, but it's the only explanation that makes sense to her or him, given that disconnection in his brain between his emotion and vision. So his vision is normal. That's why he can read a newspaper and he says it looks like my mother. His emotions are normal. He can laugh, he can cry, he feels sad, he can joke. But the connection between vision and emotion is gone. That connection is gone. Therefore, when he sees his mother, there is no emotion evoked. And when there's no emotion evoked, he says, doctor, this must be an imposter. So this is very different from the Freudian explanation in terms of the anatomy of the brain. So if you look at visual signals in the normal brain, I wish I had a drawing, but the messages go to the back of the brain, to the visual areas. Visual signals are processed. Then they go to the fusiform gyrus, where the visual signals are analyzed, further analyzed and recognized as this is my mother, this is my father, this is Joe, this is Harry the milkman or whatever, right? And then after the image is recognized, it gets relayed to the emotional centers in the brain, where there's a structure called the amygdala that performs an emotional surveillance of what you're looking at. Is this somebody I should watch out for, or is it just Joe? Is it a lion? Is it a tiger? Is it a potential mate that I should pursue? What is it? Is it something I should pay attention? So the amygdala gauges the emotional significance of the object you're looking at, which has already been recognized, quote unquote, by the fusiform gyrus, and in conjunction with other brain regions. So in this person, David, what's happened is the fusiform gyrus is itself intact, so he's still able to recognize people. He's able to recognize people and say, this woman looks like my mother. But the link to emotions is gone, so the mother does not does not produce the warm glow or terror that you would expect when you see your mother. So this delusion kicks in, and he says, my God, this must be an imposter. Now, this never made much sense to me, this explanation, because I've seen patients who have this disorder, not about their mother, but about their pet poodle. They'll say, Professor Ramachandran, this is not Fifi, it's some other dog pretending to be Fifi. Now, you try to use the Freudian explanation here. You have to start talking about the latent bestiality in all humans, or some such rubbish, right? So, we feel our anatomical explanation gets closer to the truth. Now the question is, how do you test it? So what we did was something very simple. When you and I look at anything in the world, if you look at a chair or a table, messages go to the visual areas of the brain, to the fusiform gyrus, goes to the amygdala says, and the emotional part of the brain says, big deal, it's a chair or a table, don't worry about it. Right? If you see a lion or a tiger, or indeed any familiar person, what happens is the message then goes to the amygdala says, look, watch out, it's a lion or a tiger. And then it arouses your brain, messages cascade down your autonomic nervous system, your heart starts beating faster, your blood pressure goes up to prepare your body for the ensuing action, whether it's a prey or a predator or a mate, to prepare your body for ensuing action, your blood pressure goes up and you start sweating, anticipating muscular exertion. And you can measure the sweating by putting 2 electrodes on the palm, measuring the change in skin resistance. It's called the galvanic skin response. This gives you a measure of the person's spontaneous emotional arousal when he or she looks at something in the external world. So when I take any one of you here and I show you tables and chairs and books, you don't get a galvanic skin response. You don't start sweating, obviously. And if I show you a picture of a lion or a tiger, you get a big jolt in the galvanic skin response because you start sweating. If I show you pictures of unfamiliar people you've never seen before, there's no change in galvanic skin response. You don't sweat. If you show you pictures of familiar faces, now the amazing thing is if I show you a picture of your mother, everyone here starts sweating. You don't even have to be Jewish. Okay? Now, if you do it on this patient, our patient, David, we found that chairs and tables did not produce any sweating, but nor did his mother. There was a galvanic skin response, was completely flat, showing our idea about the disconnection between visual areas and emotion centers of the brain is essentially correct, right? But even more surprising is when David's mother goes to the adjacent room, and after a couple of hours she phones him and says, "David, how are you?" David says, "Mom, how are you? It's very nice to hear from you. It's great, where have you been?" So there's no delusion on the phone. Then she comes back into the room after an hour. He says, "Who are you? You look just like my mother." So the delusion kicks in. Now, how do you explain this? It turns out there's a separate wire, to put it crudely, going from the auditory centers in the brain, auditory cortex, to the emotional centers, to the amygdala. That wire has not been cut by the accident in David. So when he gets on the phone, the voice evokes the emotions appropriate to the mother, but when he sees her in person, visual signals don't get to the amygdala, so the delusion kicks in. The reason I'm taking so much time explaining this is a powerful example of what we call behavioral neurology or cognitive neuroscience in action. And that is, you can take a bizarre, incomprehensible psychiatric syndrome, neuropsychiatric syndrome, a person thinking his mother is an imposter, and then do a very simple experiment, and reject the Freudian explanation, come up with a simple explanation in terms of the known anatomy and circuitry in the brain, and then spend 1 hour testing your model and showing that you're on the right track. Since then, there have been a number of studies on this showing that our explanation is correct. Now, let me give you one other example, which is even more bizarre, if you can believe that. Everyone here has heard of a phantom limb. In my previous book, I described phantom limbs in great detail. If you amputate an arm or a leg, the person continues to vividly feel the presence of that missing arm. And this is called a phantom limb, and we've done extensive experiments on this. For example, if a patient has a painful phantom, which is fixed in a particular position, you can put a mirror in like that on the table, and the patient puts his other hand in the mirror, looks at the reflection of the mirror, reflection of his normal hand, intact hand in the mirror. It's as though you've optically resurrected his phantom. If he now moves his real arm, the reflection of the real, it look like the phantom is moving as well. And this seems to relieve the pain in many cases. So it's not being used as a treatment for phantom limbs all over the United States and indeed the world. I'm going to wait till this noise stops. Okay, so that's about phantom limbs, but now I'll tell you about a syndrome that's even more strange, much more rare, although not that rare as it turns out, and it's called apotemnophilia. This is a person developing a desire, it started very early in childhood, an intense desire to have his own arm or leg amputated. Person desires amputation, it's called apotemnophilia. And of course, again, the Freudians got hold of this disorder and had a field day with it, and said, they came up with all kinds of explanations. Now remember, these people, in all cases in the syndromes I'm going to tell you about, the patient is completely normal or relatively normal in most other respects. So he's not psychotic, he's not mentally disturbed, he's articulate, he's intelligent, fluent in conversation, he's attentive, everything is fine. His perception is fine, his memory is fine, but he has one profound delusional fixation. In this case, the patient wants his arm removed, amputated. And from early childhood is when it begins. And sometimes they go on to get it amputated in about half the cases, and after the amputation is done, they feel much better, and any depression they had prior to amputation, they're relieved of depression. In fact, I've yet to see a case who regretted having the amputation done. So this is quite extraordinary if you think about it, loss of a limb, deliberately having an arm amputated. Why would it happen? So the two Freudian theories, one is just a wish fulfillment. In other words, not wish fulfillment, I'm take that back. One of these ideas is that it's a cry for attention. The patient wants attention, therefore he wants his arm amputated. This is completely idiotic. Why not remove the ear or the nose or something else? I mean, which is much less costly in terms of what you're losing. Why the arm, right? Another theory, Freudian theory, is the guy wants his arm amputated because he wants a big stump that resembles a giant penis. I'm not making this up. You can go and Google, and you'll find it. So all of this sounded like rubbish to me. And then I went and looked at the map of the brain, in the brain of the body surface. It turns out there's a complete map of the body surface on the brain, on the side of the brain, in a vertical strip of cortex called the post-central gyrus. So every point is represented on a point in the brain. So there's a complete map, complete representation of every body part in the brain. Now that's a sensory map. We call the Penfield map of the skin surface in the brain. Now, slightly behind it, there's a map of the joints and muscle sensation. So if you do that, the muscle and joint sensation go to that part of the brain called secondary somatosensory cortex. But then further behind in the parietal lobe, there's an area called superior parietal lobule. Pardon the jargon, but I'm not going to call it area A or area B. I'll just call it superior parietal lobule in the right hemisphere, where you construct a more abstract representation of all your body parts. So if I close my eyes, I have a vivid sense of my body, internal image of my body called the body image. That's in the superior parietal lobule. Even when I look at my hands and arms, and then I move my arms around, I dance or something like that, all of this is constructed, a dynamic, vibrant image of my body, incorporating vision, touch, proprioception, joint sense, muscle sense. It's created in the superior parietal lobule. And it turns out that the scaffolding of your body image is innately specified. It's present at birth. And we know that for other reasons. Okay. Now the sensory signals come to the sensory area of the brain, postcentral gyrus, and then they go to the superior parietal lobule, where you construct a more abstract body image using vision, hearing, all the senses to construct a body image. Now my idea was maybe this image is intact, but the arm region of this body image is missing congenitally because of a circuit malfunction, if you want to put it that way. This is an idea I had along with Paul McGeoch and David Bryan, who's a graduate student in our lab. So we decided to test this, and sure enough, when we did brain imaging, touching different parts of the body and looking at the map in the brain, the sensory map was completely normal. So the sensory signals from the hand were obviously activating nerve cells in the postcentral gyrus. So that sensory representation is normal. But when we went further back to the superior parietal lobule, the entire body was represented here, but no arm representation. That was missing. So the idea is one region of the brain, the sensory region, is getting the signals normally, but there's nowhere for the signals to go to. There's an empty region. There's nothing there. So there's this tremendous discrepancy. If you talk to these patients, a lot of the time in medicine, we are taught that just talking to the patient will clinch the diagnosis 90% of the time. If you ask them, why do you want your arm removed? Does it feel alien? Does it feel like it doesn't belong to you? They'll say, no, on the contrary, it over belongs to me. This is one of the phrases they use. Or it's intrusive, it's over present, it's getting in my way, it's intrusive, doctor. And I'll feel much happier if it weren't there. This is tremendously annoying. Now, you can see what's going on. Sensory signals are coming in, but there's no place in the brain for the signals to go to. And there's acute discrepancy. There's a discrepancy intolerance in the brain, leads to this abhorrence of the arm. When you remove it, the chap is no longer depressed and he's happy. So it's another example of this sort of analysis So what we have done is to go and take strange, odd syndromes, and you can play Sherlock Holmes, play detective, look for clues, come up with hypotheses, and then discover what's causing the problem in his brain. That's one agenda, explaining the patient's symptoms to him and showing him he's not crazy. Second agenda is so what? Okay, you explain the symptoms. Does it have any broader implications for all of us, quote unquote, normal people here? The third agenda is clinical. Can you help do something to treat the patient? Which we've done with phantom limb pain and phantom limbs. Now we did extensive series of experiments with phantom limbs and mirrors and treating phantom pain and all of that. But recently we stumbled on another observation. Just when we thought we had exhausted all the experiments possible, I was watching a phantom limb patient. So imagine I'm the patient and I lost my left arm. And my left arm is actually a phantom, a very vividly present phantom arm. And somebody goes and touches Ken here, who's sitting in the audience. And I'm the patient of the phantom limb. I'm just watching Ken's right arm being touched and stroked. Guess what happens? I feel the touch and stroke in my phantom limb, although I don't have an arm. And I'm just merely watching another human being touched and stroked. Now, how in heaven's name could that happen? Now, I was reading some papers by Giacomo Rizzolatti and listening to a talk given by him. He discovered a class of neurons called mirror neurons. And in that class of neurons, I found the explanation. I'll explain this briefly. It turns out in the front of the brain there are nerve cells called motor command neurons. So in the right frontal lobe, I have in prefrontal area and frontal cortex, I have neurons which send commands through the brainstem to the arm, through the brachial plexus, and then these commands orchestrate the sequence of muscle twitches I need to go and reach for a stone here, reach for a pen, put a peanut in my mouth, reach for a peanut, push an object, pull an object. These are all semi-skilled actions, and different neurons are there, different command neurons for different simple commands. This has been known for over 50 years, discovered by Vernon Mountcastle at Johns Hopkins, right? So these are called motor command neurons. And monkeys have them, people have them. And these are in the premotor and motor cortex. So there's a program that's used to generate the appropriate sequence of muscle twitches to execute that action. Now what here Rizzolatti found was about 20% of these neurons, 1 out of 4, 1 out of 5 of these neurons, will not only fire when I reach out and grab a peanut, but when I watch you or you reach out and grab a peanut. So they're immediately called monkey see, monkey do neurons, or mirror neurons, because they're mirroring somebody else's behavior. And they're found in humans too. So this is a very exciting, extraordinary important, because what the neuron is doing in effect is saying, is adopting other person's perspective towards that peanut. The neuron is firing, and the person is saying, the same neuron is now firing as would fire if I were to reach out and grab a peanut. Therefore, that person is now reaching out and grabbing a peanut. So it's doing a virtual reality simulation of the other human being's mind or brain, internal events in that person's brain. So now it turns out that a mirror neurons also for touch. If you touch me, if somebody were to touch me, my left, I already told you, postcentral gyrus cells fire, and areas behind it also start firing. Most of these neurons are called sensory tactile neurons because they respond to touch and they fire. There's a map in the brain of all the surface of the body. But some of these neurons, yes, you guessed it, about 10% of them, will fire when I simply watch Art here being touched. So I simply watch you being touched. The sensory neurons in my brain, just think about it. I'm watching you being touched, and the sensory touch neuron, exactly. The touch neurons in my brain are firing like crazy. So this raises an interesting question. If the sensory neuron in my brain fires when you touch Art, how come I don't feel it? You touch him, I do it. Okay? I see him being touched, and I have some empathy. If you hit him, just hit him. Okay. I feel some empathy for this, but I don't literally say, ouch, and I don't draw my arm away. Why is that? How does the brain know the difference? The answer is this input from the skin receptors going back to my brain and vetoing the output, partly vetoing the output of the mirror neuron, saying, look, you're not being touched, he's being touched. So empathize by all means, but don't literally start experiencing the touch. Empathize with his pain, but don't say ouch and withdraw. Don't confuse his pain with your pain. So I call these empathy neurons, right? But now here comes an interesting question. If this is true, how do you test it? If what I'm saying is true, then my own intact skin and receptors are going to my brain and inhibiting the output of the mirror neurons, thereby allowing me to empathize with your touch, but not literally dissolve into you or experience your touch sensations. How do you test this theory? Well, the answer is if you amputate my arm, there's no sensory signals coming in. If I watch you being touched, I should feel it in my phantom. That's exactly what we found. Not only that, what Lisa, one of my grad students, found was, Laura, what she found was, you simply inject anesthetic into the arm, temporarily paralyzing the arm into the brachial plexus, temporarily anesthetizing the arm becomes numb, and I watch you being touched. I feel it in my anesthetized arm. So the only thing that's separating your mind, your sensorium from my mind, is the skin, and there's airwaves between us. Otherwise our brains are all hooked up together, like the global worldwide net or Facebook. This is already invented by evolution several 100,000 years ago. And so hence the importance of mirror neurons. I also argue in the book that the mirror neurons are important for, as I said, for adopting your point of view on a problem or on some task you're engaged in. Now, why is that important? It's important for imitation. You say imitation, well, who cares about imitation? I mean, that's mockery. It's actually insulting. But not at all. Actually, imitation is the dawn of culture. Because if I watch you doing a skill, I learn at once how to do it. And to do that, I need to put myself in your shoes and look at the world from your point of view and see you as an intelligent, I don't know about intelligent in your case, but see you as an intentional, purposeful being conducting a certain task in order to imitate. Now you say, well, why is that important for culture? Well, think about a polar bear evolving a fur coat. It'll take several 100,000 years of painful, laborious natural selection to evolve a fur coat. A human child, an early hominin child, like early Homo sapiens or Homo erectus, watching his mother hunt and skin a polar bear, can learn it in one trial, in one generation. Or the peers, other neighboring polar bears, neighboring children, can learn to skin a polar bear in one generation. So what took hundreds of thousands of years can be done in one generation by imitation. Hence the importance of imitation. So hence you see the dawn of culture and the dawn of civilization, all because of this group of neurons called mirror neurons. Now remember, even monkeys, lower primates have mirror neurons. It's not, obviously it's not enough for imitation. You need it to develop a certain level of sophistication, which happened in humans, and develop connections with other brain regions. Once that happened, imitation learning could take off in human evolution, thereby leading to a lot of what we call civilization. That's about mirror neurons. Last thing I want to tell you about is another odd condition, which is even more common, I should say, which is common. It's called synesthesia. And many of you may have heard of this. It was first described by Francis Galton in the 19th century. He was a first cousin of Charles Darwin. He noticed that some people in the population were otherwise completely normal. Galton thought it was very rare, one in 1,000 or something like that. But we have shown 1 in 50 people have synesthesia, this condition called synesthesia. That is, these people have the following quirk. Every time they see a particular number, they see a particular color. So 5 is red, 6 is blue, 7 is yellow, 8 is indigo, 9 is chartreuse, so on and so forth. Or every time they hear a tone, it's a particular color. F sharp is blue, C sharp is green, and so on and so forth. Now Galton published this in Nature. He also pointed out it runs in families and may have a genetic basis. Now since then it's also been shown, dozens of times, hundreds of synesthetes have been described. It's been shown that not only is it heritable, it may have a genetic basis like Galton said, it's also 7 or 8 times more common in artists, poets, and novelists. Now why would that be? Now first of all, what causes synesthesia? Most people thought this was just, the people were just crazy. This is a common reaction in science. When something doesn't make any sense, it's regarded as an anomaly and brushed under the carpet, something that a lot of my colleagues are very prone to doing. So this is a form of denial. And a lot of the time these are of course false alarms, anomalies, but every now and then an anomaly is an authentic anomaly that can lead to a gold mine, and I believe that synesthesia is one such example. So one of the theories that's been around is these people are just crazy, but let's set that aside, even if it's true. Another theory is they are acid junkies and potheads. And sure enough, this is much more common in Berkeley than at UCSD. So there might be some truth to that idea. But the third view is that these people are being metaphorical when they say F sharp is green or blue. Just like you and I say cheese is sharp. Cheese is soft, it's not sharp. So why do you call it sharp? Then you say, well, it's a metaphor. But why do you, it's the circularity there. Why do you use a tactile metaphor, soft, to describe a gustatory sensation, or sharp to describe a gustatory sensation? These are called synesthetic metaphors. Now the problem with the explanation, saying synesthesia is just metaphor, is in science you can't get very far by explaining one mystery with another mystery. If you say synesthesia, well, that's just a metaphor. Well, what the hell is a metaphor? We don't have any idea how metaphors are represented in the brain. All you know is a metaphor involves linking two seemingly unrelated things, right? So what I'm going to do in the next 5-10 minutes is to say the opposite, is to say that synesthesia is an abnormality of sorts, or you can call it a gift, that's seen in some people, and it's not, but you can study it and establish its neural sensory basis in the brain, and that in turn gives you an experimental handle, part of the metaphor, experimental handle to understand elusive aspects of the brain, such as metaphor. So you're going to go from this odd phenomenon, synesthesia, all the way to metaphor in Shakespeare. How do you do that? Well, the first thing we wanted to show was it's a genuine phenomenon. We did a number of experiments to show these people are not crazy. They literally see red when they see 5, see green when they see 3, see blue when they see 2, and so on and so forth. It's different colors for different people. Okay. Ed Hubbard and I were looking at brain atlases, and we were struck by the fact that in all of us here, not just in synesthetes, the color area in the brain, which is processed in the fusiform gyrus, just like faces, the color area in the brain, where colors are processed, is right next to the number area in the brain, where the visual appearance of numbers is processed. They're almost touching each other. So we said, what's the likelihood that the most common form of synesthesia is number-color synesthesia? And these two areas are sitting right next to each other in the brain. Maybe in some people there is some accidental cross-wiring. And because of this accidental cross-wiring, every time he sees a number, not only is a number neuron excited, but there's a co-activation, there's a cross-activation of a color neuron. So every time he sees 5, he sees red. Now, we tested this by doing brain imaging. We show normal people just black and white numbers. Number area lights up in fusiform gyrus. You show colored numbers, number area and color area lights up. If you show synesthete, black and white numbers, number area and color area light up, showing that our explanation is correct. Since then, there have been other studies confirming this. Now, the big question is, why does it happen? Why do some people have synesthesia? Why do the areas get cross-wired in these people? The answer comes from realizing that in the fetal brain, in early infancy, all the brain regions are connected to each other. This is overstatement, but it's roughly correct that there's a gross excess of connections and redundancy of connections across connecting even far-flung regions of the brain. And then there are pruning genes that prune away the excess connections to create the modularity that characterizes the adult brain. Specialized regions characterize the adult brain. So in other words, in the fetus, the color area and number area were in fact possibly connected to each other. Similarly, hearing and color. And then the pruning genes come along and whittle away or prune away the excess connections. If there's a mutation in this pruning gene, this pruning fails to occur between adjacent brain regions, and you get an excess connections between the number area and color area, you get number color synesthesia. So long as that gene is expressed selectively because of transcription factors only in the fusiform gyrus. Supposing the same gene is expressed throughout the brain, you get excess connections throughout the brain. Now that gives you the very important clue. Remember I said early in the talk that synesthesia is 8 times more common among poets, artists, and novelists. Why would that be? And what do poets, artists, and novelists have in common? They're very good at metaphor, linking seemingly unrelated things. Like it is the east and Juliet is the sun, as Shakespeare said. So when you say Shakespeare is the sun, you don't say, well, she's a glowing ball of fire. Actually, schizophrenics say that. No, I'm serious. They take metaphors literally. But most normal people would say, well, she's radiant like the sun, she's glowing like the sun, she's warm like the sun, so on and so forth. Your brain forms all the right links. Now, if you assume that concepts and ideas are also in different brain regions, represented in different brain regions, then if you have synesthesia gene expressing itself throughout the brain, producing excess connections between different brain regions, you're going to confer on that brain a propensity towards linking seemingly unrelated concepts and ideas, hence a propensity towards creativity and metaphorical thinking. And that's the hidden agenda of the gene, I'd argue. That's why the gene has been conserved and seen in fact, we see 1 out of 50 people as a synesthete. It's not rare. And if a gene is useless, it would have been weeded out hundreds of thousands of years ago by natural selection. So what it does is, in some outliers in the population, it's expressed more diffusely, and it makes them more metaphorical and creative. Some people, it's expressed selectively in the fusiform, and you see number color synesthesia, which is useless but is not harmful. But it's a penalty you pay for the fact that there are some outliers who have this gene being expressed diffusely, making people more creative and metaphorical. But when I give these talks, people often ask me, in fact the other day up in LA somebody asked me, well, if this gene is that good, how come we all don't have it? Right? It makes people creative. Well, that's a silly question because in evolution things take time. Maybe another 200,000 years from now, everybody will have the synesthesia gene. That's one answer. But another possible answer is you don't want everybody to be metaphorical and creative. You need engineers. Also, if a neurosurgeon is performing surgery on your brain, you don't want him getting metaphorical or creative on it. So this smacks of group selection, you have to be careful. But you do need a heterogeneity in the gene pool, a diversity of spectrum of human abilities, and that's what this gene seems to be doing. So I think I'm going to conclude by saying the book is full of examples of this kind where you begin with an anomaly or curious observation like synesthesia. Here's an example of people seeing colored numbers. Who would have thought you can start with this? And people are now starting to clone the gene. You can go from the gene, synesthesia gene, to the phenomenology, people seeing colored numbers, and then exploring the psychology and psychophysics of this, showing it's a real phenomenon. These people are not making it up. Then to neuroanatomy, studying the brain anatomy, all the way to creativity, Shakespeare, and metaphor. All starting with this quirky observation on these people with synesthesia. And that's what we've done with each of these syndromes. And we also touch on the question of human uniqueness and parts of the human anatomy, human brain anatomy, which make us different from the great apes. And our brains have specialized regions which make us different from the great apes. So let me conclude by saying saying thank you.
Yeah, about the synesthesia with the colors and the numbers. Here in the West, we use Hindu-Arabic numbers, but other cultures use different visual representations for numbers: Roman numerals, Japanese, Chinese, and so on. Uh, have enough Hello. Have enough tests been done with people from other cultures that they don't use Hindu-Arabic numbers, or maybe they've never seen them. Maybe they use Roman numerals, or maybe or Chinese or some other kind of representation, or maybe even illiterates. Maybe they don't have a written component to their language.
That's a fascinating question. We haven't studied illiterates, but we've looked at other languages, and sure enough, they have synesthetes just as commonly as people speak. I'm glad you said Hindu-Arabic, by the way, because most people just say Arabic. And the number system comes from India, as you know. A bit of jingoism there. But to answer your question, yes, you do. Now, when I say this synesthesia is hardwired or genetic, what I mean is a propensity to link graphemes, arbitrary symbols, to certain colors. That propensity is innate, not which particular symbol you choose. That obviously depends on what your native language is and what your graphemes are. It'd be interesting to see if you take completely illiterate cultures where they don't have symbols for different numbers, what would happen? Would they develop synesthesia for arbitrary shapes, like triangles and squares and that sort of thing? Nobody's ever studied that. Good question.
Roger, this gentleman.
Question.
Have there been any studies with synesthesia where normal people are placed under hypnosis and are conditioned to see color and number together, and then both centers in the brain light up?
I see. That particular experiment hasn't been done, but people have tried overtraining normal people to see if you can get them to become synesthetes. That's not worked either. But they've taken synesthetes and trained them on completely new, novel shapes and colors. And my student Dave Brang has done that, and they become synesthetic for those new symbols and colors. But in terms of things like, people have asked me, what about meditation, what about hypnosis? It's not been tried on any systematic basis. There may be a study out there, but not that I'm aware of. Roger.
There's a limit to how far we can get this thing to go, but if you would get a bit closer, perhaps.
Yeah.
And maybe you could just wander up here.
The mirror neurons go. No. Has Madison Avenue shown any interest in the chair?
I'm sorry?
In the mirror neurons, has Madison Avenue shown any interest in?
Oh, so the question is about whether the advertising world has got.
I'm sure
started
that
using mirror neurons. Yeah.
they haven't approached me yet, but I'm sure.
No, it wasn't about you, Raman. It was about
No.
the industry in general.
Well, it could be. I mean, if they were,
Sure.
could fund some research projects.
Are there any Stephen Hilliard?
Oh, but by the way, I wanted to say I didn't mention autism, which is not relevant to your question, but yeah, sorry.
Are there any Stephen Hilliard type electrophysiologic manifestations of the synesthetic?
Yeah, my student Dave Brang has been studying that, and you should email him. It's a long story, but yes, there has been, and also on mirror neurons too. If you do EEG, there's a component of EEG called mu wave suppression. What happens when I move and grab a peanut, my mu wave gets suppressed in my EEG. But if I watch you grab a peanut, my mu wave gets suppressed. So this is supposed to be the correlate. We have suggested it's a correlate of mirror neuron activity in humans. It's a simple way of monitoring their activity.
So let me ask you a question, and then we'll go over there. But we've done 3 meetings now called Beyond Belief at the Salk Institute. We've talked about science, religion, reason, survival, God, all sorts of things. We've had Richard Dawkins there, all sorts of
And does everybody here know about it? Because it's an excellent thing to.
I think, yeah, Neil deGrasse Tyson was there. But in the book, you actually mention your sense of what you think, what your perspective is on the whole notion of God, religion, Spinoza's God, and so on and so forth. Could you gloss that a little bit?
Oh boy.
That's a question for Art. Okay.
You can do that. You can.
Well, there's 2 separate questions. One is in terms of belief in God, and religious sentiments and religious feelings obviously arise in the human brain. And we know people with temporal lobe seizures have intense religious mystical experiences, so somehow bits of the temporal lobes are involved. So that's about the subjective experience of mystical union with God or mystical union with the cosmos. But that's the scientific, physiological, neurological question. But there's a metaphysical question about Atman and Brahman and universal consciousness and how that's connected with the spirit and all that. But that's really outside the scope of my book or indeed of this presentation.
About the example of David, where he
The cop grab patient.
recognized the identity of his mother visually, but did when she went out of the room over the phone.
Yes.
If she's standing there and starts nagging him,
Yes.
what is his perception of her?
That's a good question. And what tends to happen is we've done it a few times. And what happens is the visual delusion kicks in. It overrides the auditory. And that you would expect that because we are highly visual primates, and we're constantly weighing giving weights to different cues, sources of information. And we primates give more weight to visual cues. So for example, tomorrow if somebody sounds like you comes here but looks utterly different and starts talking, then I won't confuse him with you. Right? But on the other hand, if you come here with a pharyngitis, I'll still recognize you and say, well, you have a sore throat. So that's because we rely much more on visual cues. So it's a good question, though.
There's a question behind you, Ramach.
Rama, in chemically induced altered states of consciousness, the most common one being alcohol-induced, is there people often feel they're more creative when they're high. Defining creativity as being a hyperlinked state of the modules of the mind, is there any way in which sort of correlating something else you said, which is that there's a repression that goes on from other inputs. The correlation or the connections between the different modules, not only are they trimmed neurologically, neurophysiologically, are they inhibited in a way that we talk about disinhibition or release? Could there be a real physiologic basis for when the creativity linked to various kinds of altered states?
Well, I think that intuitively that seems likely. We've seen patients, neurological patients, for example, develop temporal lobe seizures, suddenly writing poetry which they could never do before. And I had one patient write to me saying he was never interested in poetry. Suddenly he started writing poetry and he wrote a poem about me. And it wasn't very good. I mean,
that's the hypergraphia.
Yeah, exactly. But it was reasonable. It was rhyme. It had rhyme and all that. Sorry. Okay. So it was reasonable. And then there's experiments by Alan Schneider in Australia, where he zaps portions of the brain, especially the frontal regions of the brain, with TMS, transcranial magnetic stimulation, temporarily silencing a part of the brain. And during that period, the person starts drawing beautifully, or doing numerical calculations much more effectively than normal people, or than he could before the stimulation. So such things are possible, but they've never been studied on a systematic basis. People often ask me, is it true that, can you unleash hidden powers in all of us? Is it true that we only use 10% of the brain? And I say it's true in Texas, but I don't know about the rest of the world.
Dennis, you had a question, I think?
Yes. Toward the end of Nobel laureate Francis Crick's life, you and he were having a discussion about the concept of a unity of conscious experience, and he gave you a sly conspiratorial wink, as you pointed out in your book, about some part of the brain that he referred to as the claustrum.
Yeah.
Could you elaborate on Dr. Crick's
cryptic remark?
Yes.
Well, Francis was obsessed with the idea of mapping function onto structure, because he was immensely successful in doing that with DNA. And he was trying to achieve the same thing with consciousness and qualia and brain function. And he was never afraid to speculate very boldly. And it turns out there are some brain structures, such as the claustrum, which very little is known about. There's some single unit recording by Simon LeVay, the Salk, but by and large we know almost nothing about it. There are no human cases with selective damage to the claustrum on two sides. But we know its anatomy is connected to large numbers of brain areas. So he, in a very quote-unquote simple-minded manner, says we have this sense of unity of conscious experience. Maybe it's not an illusion. Maybe this structure, which is connected to everything else in the brain, plays a vital role in aspects of conscious experience. So it was just a throwaway remark, but who knows? He's been right often enough. Right? But I think what's missing in brain research is, in the case of DNA, the functional logic of heredity and maps on beautifully to the complementarity of the helices, right? So the complementarity of offspring and parent, there's an analogy between that and the complementarity of the DNA strands. So that was the intuitive leap which helped them solve the greatest mystery in biology at that time. In fact, you could say biology was born at that moment when they saw that analogy and grasped that analogy, and then subsequently went on to tackle the genetic code. There's been no such tremendous insight in neuroscience where you say, look, the functional logic follows beautifully from these neural structures. There have been lots of correlations, but that's the pre-DNA era where he said, if you zap the chromosomes, you get mutations, so something interesting is happening in the chromosomes. That's the stage we're at in neuroscience.
I've been scrambling through the book here for this. There was a very funny story on Capgras, where you were woken up in the middle of the night by some attorney from England.
Yes.
Right? Do you want to tell the story?
Why don't you tell it?
All right. So this person had been in a car accident, seemed to have brain damage, seemed to imagine, having read your literature, that he had Capgras syndrome, and he was trying to sue the people that injured him in the car crash because he could no longer recognize his wife. I mean, he thought his wife was an imposter or something. And
well, he said that she looked like his wife.
Looked like his wife, but yeah.
But she didn't look familiar anymore.
And you said, in typical Ramachandran problem, what's the problem? If Mr. Dobbs finds that his wife looks like a new woman every time he sees her, he should find it personally attractive, perpetually attractive. This is a good thing, not bad at all. We should all be so lucky.
My wife's sitting right here, so
showing great restraint.
Okay, so this one over here. All right.
Dr. You've talked about examples when you're doing experiments. You show pictures of a lion, a human, a car. But our mind knows that that's a book with a picture of a car, a lion. What role is video taking in terms of your sense of reality now with experiments? And if there is more of a connection of reality, could therapies in the future be primarily video-based?
Oh, yeah, that's a very good question. In the case of Capgras, for example, we showed photographs of his mother or photographs of his parents. The experiment worked fine with just the photographs. But I bet if you did it with real people, you get a much bigger and stronger effect. Short answer to your question is the more the realism in the stimulus, the more clearer your results. But often it's impractical to do that. So for example, with the mirror treatment for phantom limbs, it's the realism of the reflection, the fact that it looks so utterly real, that seems to help. But he knows at some level that it's not real. That seems not to matter, because the brain has multiple layers of belief and it's modular in its organization. So what one module is performing and signaling, saying that's a real arm, cannot be vetoed by another module saying, that's stupid, that's just a mirror, it's not your real phantom arm, it's not your real arm. So it's a consequence of modularity of brain function.
So do you want to comment a little bit with the piece I read out at the beginning about how you use low-tech methodologies and so on? You also then go on to say, look, I'm not a Luddite. If this stuff works, I use fMRI as well and so on. But there's a cautionary note in there, isn't there, about how much information you can really get from fMRI, and there's been lots of papers written about recently about the sort of the voodoo aspect of it. So.
Well, I think using high-tech stuff, actually using very low-tech things, actually forces you to be more ingenious in using it. This is true even in medicine. If you don't have, in the old days before CT scan and MR, you had to be an astute clinical neurologist, elicit signs and symptoms, and ask the patient, get his history in great detail, and often figure it all out just from that. So first of all, it gives you an intensely, you have to be much more resourceful in the absence of high-tech equipment, right? The other thing is that if you use, it's like giving a 6-year-old child a knife. It just goes and keeps cutting everything in sight. So this is like an FMR machine. I mean, it's not goal-directed. The problem then becomes technique. In other words, the research should be problem-driven, not technique-driven. And that's a serious problem in this field. And lastly, I'm also lazy. I don't want to sit down and learn all the technology. If I can use a simple technique, why not use the simple technique?
Dennis, it's 8 o'clock. Do you want to have people sign books? Thank you, Ramachandran.